So, hi, I'm Jo Mauskopf, and I'm reading actually to substitute for someone else who wasn't able to read. And I'm reading a few extracts from The Economist. And these come from the science and technology section of The Economist, and they're mostly about medicine and COVID and infectious diseases, and I hope you'll find them interesting. The first one is called Science After the Pandemic. COVID-19, this is the subheading, has brought together medical technologies that will transform human health. The first virus to have its genome read was an obscure little creature called MS2. The 3,569 RNA letters it contained were published in 1976, the hard-won product of some 10 years' work in a well-staffed Belgian laboratory. The SARS-CoV-2 genome, almost 9 times longer, was published just weeks after doctors in Wuhan first became concerned about a new pneumonia. That feat has since been repeated with getting on for 1 million different samples of SARS-CoV-2 in the hunt for fearsome variants like the one ravaging Brazil. Within weeks of its publication, the original genome sequence became the basis for the vaccines that today are stymieing the virus whenever supplies, politics, and public confidence allow. It is hardly remarkable that medical science has moved on since 1976, but the COVID-19 pandemic has brought the sharp joy of seeing decades of cumulative scientific progress in sudden concerted action. The spate of data, experiments, and insights has had profound effects on the pandemic, and indeed on the future of medicine. It is also an inspiration. Around the world, scientists have put aside their own work in order to do their bit against the common foe. Jealously guarded lab space has been devoted to the grunt work of processing tests. COVID-19 has led to some 350,000 bits of research, many of them on preprint servers that make findings available almost instantaneously. The basis of all this is the application of genetics to medicine in a systematic and transformative way, not just in understanding the pathology of diseases, but in tracking their spread and curing and preventing them. This approach could underpin what is becoming known as natural security, the task of making societies resilient in the face of risks stemming from their connection to the living world, whether because of disease, food insecurity, biological warfare, or environmental degradation. The application of genetics to medicine partly reflects huge rapid gains in efficiency. Reading the DNA in a human genome cost $10 million in 2007. Today it takes less than $1,000 and a fraction of the time. Coupled with ever better ways of synthesizing and editing genes, this has enabled cleverness little short of the miraculous. Before the pandemic, these trailblazing techniques were not much talked about beyond the laboratory. Having shown their mettle against a brand new disease, they have burst out into the open. Take the vaccination technology rapidly developed by Moderna of America and BioNTech of Germany, building on years of patient and often unsung work on RNA, a store of genetic information. It is remarkable that you can simply instruct the body's cells to make the viral protein you have designed to prime the immune system. The RNA vaccines are testament to the insight of Eddie Cantor, a comedian, that it takes 20 years to become an overnight success. With this proof of concept, the investments of companies that have worked hard on RNA may now pay off. To some extent, RNA medicines divorces form from function. An RNA vaccine against any disease is a message written in genetic code. A vaccine against malaria or some form of cancer can be made in the same way and with the same equipment as a SARS-CoV-2 vaccine. If this provides a platform for getting cells to do all sorts of specific things and to desist from others, as it promises to, medicine will become both more powerful and more personal. Therapies tailored to rare, even one-off genetic abnormalities should become routine. The pandemic has also demonstrated the value of gene sequencing technologies. Observing SARS-CoV-2 as it mutates is essential if the world is to understand and defend itself against dangerous variants. Should COVID-19 become endemic, as is likely, sequencing will become the basis for developing regular booster shots. More broadly, routine sequencing is one of the best ways of knowing what is out there. Companies have done brilliantly in producing powerful sequencing systems for trained technicians. Now the world needs cheap, ubiquitous and reliable systems that can be used in the prison sick bay or the rural health center, on the farm, or at the town's sewage works to act as early warning systems for the spread of pathogens. Another area of work is where the pandemic has revealed a gap. Even today's progress has yet to produce a small molecule antiviral to combat SARS-CoV-2. A focus for natural security should be drugs aimed at the viral families most likely to cause trouble in the future. This is not something that the market will support on its own. New mechanisms that involve governments will be needed, such as funds for R&D and trials, and to buy stockpiles of medicine. Similar approaches should be used for the looming threat of antibiotic-resistant bacteria. These innovations will have big consequences. General-purpose RNA medicine asks new things of firms and regulators, as do other platforms, including some forms of gene therapy. Regulators will need to take advantage of the fact that, say, a malaria vaccine and a SARS-CoV-2 vaccine are both made on the same platform by streamlining approval for them, while continuing to ensure safety. Drug firms will have to adapt, as some chronic conditions may, in effect, be cured. Many are used to concentrating on the long-lasting afflictions that most trouble the rich world: heart disease, cancer, metabolic disorders, neurodegenerative conditions, and the like. If drug development is more targeted on instructing cells what to do, rather than finding novel molecules against specific programs, some of the know-how on which old-style pharma is based will be less relevant. Firms will need new pricing models and a new focus to their research. Natural protection. Technology will not in itself thwart pandemics. That goal also requires systems and institutions which use technology broadly and wisely. Without good systems, great technology will often provide only mediocre results, as it has in many COVID test and trace programs. but with the pandemic has shown that biomedical science has the tools and the enthusiasm to improve the world. The world was now built on both. Now, this next piece I'm going to read is showing how we are all partly Neanderthal. And I thought that might be interesting. COVID and genetics, a mixed blessing. And then the subheading is, DNA inherited from Neanderthals can make people vulnerable to COVID-19 or resistant to it. As best scientists can tell, Neanderthals died out about 40,000 years ago, but they did not vanish from the earth entirely. In the past decade, it has become clear that Neanderthals mated with the ancestors of modern humans, and that at least some of those unions produced viable offspring. The upshot is that almost half of the Neanderthal genome still survives, scattered in small quantities among almost all modern people's DNA. The exception is in those with mostly African ancestors, for Neanderthals seem never to have lived in Africa. Such genes have been associated with everything from hairiness to fat metabolism. Many seem to be related to the immune system and to affect the risk of developing diseases, including lupus, Crohn's, and diabetes. A pair of recent papers suggests COVID-19 belongs on that list as well. Two long sections of DNA, both inherited from Neanderthals, appear to confer resistance or susceptibility to severe COVID-19, depending on which is present. The work was led by Hugo Seeberg and Svante Pääbo at the Max Planck Institute for Evolutionary Anthropology in Leipzig, whose researchers pioneered the study of Neanderthal DNA. Their first paper, published in Nature in September, described one Neanderthal DNA string known as a haplotype that is associated with a higher risk of serious illness. Having one copy of the haplotype, which is found on the third of the 46 chromosomes possessed by humans, doubles the chances of a trip to intensive care. Those unlucky enough to possess two copies, one from each parent, face an even higher risk. That genetic bad luck is not evenly distributed. The gene sequence is most common among people of South Asian descent, with 63% of the population of Bangladesh carrying at least one copy, and among Europeans, where the prevalence is about 16%. As expected, it is virtually absent from Africa. More strikingly, it is also very rare in large swaths of Eastern Asia. Exactly what the haplotype does is not clear. One gene within it encodes a protein that interacts with the cellular receptors that the SARS-CoV-2, the virus which causes COVID-19, uses to enter cells and hijack them. The haplotype is also thought to be involved in the production of signaling proteins called cytokines that help to regulate the immune system. An overly aggressive immune response is one mechanism by which COVID-19 kills. On the other hand, some of those cytokines protect against cholera. The researchers speculate that may be why the haplotype is common in Bangladesh and India, where cholera has long been a problem. And there is evidence that even as evolution has been boosting the haplotype in some populations, it has been working to remove it from others. The frequency differences between South Asia and East Asia are so dramatic that we cannot help but suspect that past selection is responsible, says Dr. Parbo. The second study, published this week in Proceedings of the National Academy of Sciences, concerns another Neanderthal haplotype found on chromosome 12. Its effect is protective, though it is less potent. Having a single copy is associated with a 22% lower chance of critical illness. This helpful sequence is more well-traveled than the harmful one. It is present in every part of the world except sub-Saharan Africa. Between 25% and 35% of the population of Eurasia carry at least one copy. In Vietnam and eastern China, more than half the population are carriers. It also exists at much lower rates among American populations of mainly African descent, many of whom will have some more recent Eurasian ancestry as well. Scientists have a better idea of what it does, for it was known to researchers even before the news of its Neanderthal origin. The haplotype hampers the spread of RNA viruses, of which SARS-CoV-2 is one, by driving cells infected with them to self-destruct quickly. It is known to provide at least some protection against West Nile virus, hepatitis C, and intriguingly, SARS-CoV-1, which caused the SARS outbreak that began in 2002. Once again, the hand of natural selection is visible. The genes in chromosome 12 haplotype are found in other mammals and have been lost several times in other species. That hints that carrying them comes at a significant cost, leading them to be removed if they're not being heavily used. That they are common in most human populations suggests RNA viruses have been a thorn in humanity's side for much of its evolutionary history. The researchers hope that their work might help shed light on why some countries and some populations within countries appear to have been hit harder by COVID-19 than others. They point out, for instance, that Britons of Bangladeshi heritage suffer severe COVID-19 at roughly twice the rate of the general population. But disentangling the effects of DNA will be tricky. Age, obesity, and sex, among other things, all influence the severity of COVID-19. Comparisons between countries are complicated by definitional differences and the difficulty of performing accurate counts, especially in poor countries. Despite the prevalence of the harmful haplotype, the official COVID-19 death rate in Bangladesh is just 5.1 per 100,000, an order of magnitude lower than in countries where the haplotype is much rarer. Still, a reminder that genetics matter, alongside those other factors, is still useful. If COVID-19 becomes an endemic disease, as seems likely, cheap gene sequencing may in future help doctors assess which patients are likely to be vulnerable to its worst effects. Understanding the mechanisms by which genes confer resistance or susceptibility may help with the search for drugs. And history suggests that SARS-CoV-2 is unlikely to be the last novel coronavirus to make the jump to humans. If some populations are likely to be more vulnerable than others, that is worth knowing for the next time. Okay, now I'm going to read a short piece about why you should wear your masks. Face masks and COVID-19, helpful humidity. And the subtitle is, as well as filtering air, face masks may boost the immune system. Face masks help reduce the spread of SARS-CoV-2, the virus that causes COVID-19. Several studies have reported the more surprising finding that even if wearers do become infected, their disease is usually milder. Now, Joseph Courtney and Ad Bax, a pair of researchers at the National Institutes of Health in Bethesda, Maryland, think they may have worked out why. As they report in the Biophysical Journal, it comes down to humidity, the immune system, and the protective powers of mucus, of which I have a lot because of the allergies at the moment. At first chance, there may not seem much of a mystery to unravel. Masks cut the number of infectious particles entering the nose and mouth. One might expect, therefore, that severe disease will be less likely. But it is not so. One vital factor which predicts disease severity is how far viral particles make it into a person's lungs. Cheap cotton face masks struggle to block the smallest aerosols, which are the most likely to penetrate deeply. Dr. Courtney and Dr. Bax wondered if something else might explain their protective effect. One of the body's first lines of defense against airborne pathogens is known as the mucociliary clearance mechanism. Sticky mucus in the nose and respiratory tract snares viruses and bacteria. Little hairs, known as cilia, push the mucus into the throat. From there it is swallowed, and potent stomach acids destroy the invaders. But this mechanism relies on the relevant body parts staying moist. That is harder in winter, for as air gets cooler, its capacity to hold water falls. Lower humidity tends to dry out the respiratory tract. This is one reason why many upper respiratory viruses, such as influenza, flourish in the winter. Dr. Courtney and Dr. Bax speculated that masks may help keep things damp. They reasoned that as a person exhales, water vapor would condense on the inside of a mask. Then on inhalation, dry air passing through the mask would collect the deposited water and bring it back into the respiratory tract and lungs. That could be handing a mask wearer's immune system a significant advantage. Keen to test the idea, the researchers checked several masks at 37 degrees centigrade, 22 degrees centigrade, and 8 degrees centigrade. They breathed into a sealed box packed with sensitive instruments and worked out humidity levels in their upper respiratory tract. They found that although all masks increased humidity levels to some extent, the heavy cotton mask did best of all. In the hottest room, it boosted the relative humidity of inspired air by over 50%, compared with breathing without a mask. In the cold room, that number rose to 300%. The other masks recorded figures of between 150% and 225%. That suggests that besides filtering out at least some of the viral particles floating about in the air, masks keep a person's snot levels healthily high. So keep wearing your mask. Okay, for my final reading, I'm going to move away from humans, from pre-humans, Neanderthals, to a different animal species altogether. And this is on the entomology, swarm immunity. We're staying with the concept of vaccines, however. Honeybees run vaccination programs too. An old saw has it, there is nothing new under the sun. but it may still come as a surprise that human beings are not alone in having invented vaccination. Work just published in the Journal of Experimental Biology by Guy Harwood of the University of Illinois in Urbana-Champaign confirms that honeybees got there first. It also suggests that they run what look like the equivalent of prime boost childhood vaccination programs. Being gregarious, honeybees are at constant risk of diseases sweeping through their hives. Most animals which live in crowded conditions have particularly robust immune systems, so it long puzzled entomologists that honeybees do not. Indeed, they actually possess fewer immune-related genes than most solitary bees. Part of the answer, discovered in 2015, is that queen bees vaccinate their eggs by transferring into them, before they are laid, fragments of proteins from disease-causing pathogens. These act as antigens, which trigger the development of a protective immune response in the developing young. But that observation raises the question of how the queen receives her antigen supply in the first place, for she subsists purely on royal jelly, a substance secreted by worker bees, which are at the stage of their lives, which precedes the period that they spend flying around foraging for nectar and pollen, when they act as nurses to larvae. Dr. Harwood therefore wondered if the nurses were incorporating into the royal jelly they were producing fragments from pathogens they had consumed while eating the victuals brought to the hive by the foragers. To test this idea, he teamed up with a group at the University of Helsinki in Finland, led by Heli Salmela. Together, they collected about 150 nurse bees and divided them among 6 queenless mini hives equipped with broods of larvae to look after. Instead of nectar, they fed the nurses on sugar water, and for 3 of the hives, they laced this syrup with pine bacillus larvae, a bacterium that causes a hive-killing disease called American foul brood. In this case, to stop such an infection happening, Dr. Harwood and Dr. Salmela heat-treated and so killed the pathogens in advance. They also labeled the dead bacteria with a fluorescent dye to make their subsequent fates easy to track. And sure enough, fluorescent microscopy confirmed that fragments of pea larvae were getting into royal jelly secreted by those bees which had been fed with the laced sugar water. Moreover, examination of this royal jelly revealed elevated levels compared with royal jelly from bees that had not been dosed with pea larvae of an antimicrobial peptide known as defensive 1. This substance is thought to help bee immune systems fend off bacterial infections. All told, these findings suggest that nurse bees are indeed, via their royal jelly, passing antigens on to the queen for inoculation into her eggs. They also mean, because larvae too receive royal jelly for the first few days after they hatch, that the nurses are inoculating their larval charges as well. Each infant bee is therefore being vaccinated twice. Whether this is simply a belt and braces approach or is actually the equivalent of a prime boost human vaccination in which the second dose multiplies the effect of the first remains to be determined. But either way, it seems protective, not so much herd as swarm immunity. So with that, I'm going to finish for today. Thank you for listening, and I hope you've enjoyed some of the science as I have.